Welcome to the deep dive. Today, we're going to be taking you on a journey
Oh, cool.
into the microscopic world
Okay.
of hybridoma cells. These tiny but mighty factories that crank out monoclonal antibodies. You know,
Right.
those targeted molecules used in everything from disease diagnostics to
Yep.
cutting-edge cancer therapies.
Oh, wow.
Our guide for this deep dive
Right.
is a resource from Assay Ginny
Hmm.
titled Passaging Hybridoma Cells, a detailed guide.
Interesting.
And trust me, it's packed with fascinating insights. But before we get into the nitty-gritty of caring for these cells,
Yeah.
let's start with the basics.
Sure.
What exactly are hybridoma cells?
So,
And why are they so crucial?
hybridoma cells are actually a bit of a scientific marvel.
Okay.
They don't exist naturally.
Yep.
They're engineered in the lab by fusing two different types of cells.
Okay.
Imagine combining the skills of a master antibody craftsman,
Okay.
a B lymphocyte,
Right.
with the tireless energy of a machine,
Yeah.
a myeloma cell.
Oh, wow.
The result,
Yeah.
a hybridoma cell that can produce a very specific antibody
Okay.
and just keep replicating,
Right.
basically giving us a constant supply of that antibody.
So they're like tiny,
Exactly.
customized antibody factories.
Yeah,
It's incredible to think that we can engineer cells like this.
it is. It is.
But like any complex system,
Right.
they need the right care to keep those antibodies flowing, right?
Absolutely.
Okay.
One of the most important aspects of hybridoma cell care is a process called passaging.
Okay.
You can think of it like repotting a plant that's outgrown its pot.
Got it.
If hybridoma cells get too crowded,
Yeah.
they start competing for nutrients in space,
Right.
and their antibody production can suffer.
Okay.
Passaging ensures they have the room and resources to thrive.
It makes sense.
Yeah.
So how do we know when it's time to repot these cells, so to speak?
There are a few key indicators we look at.
Okay.
First, cell density.
Mm-hmm.
Ideally, you want to passage them when they reach a density of around 500,000 to 1,000,000 cells per milliliter.
Wow.
It's like making sure there's not a huge line at the cellular buffet.
I like that analogy.
Yeah.
Okay, so then,
Second, confluency,
okay.
which basically means how much of the available space the cells are taking up.
Right.
For hybridoma cells in suspension cultures,
Mm-hmm.
where they're floating freely in liquid,
Okay.
around 70 to 80% confluency
Okay.
is a good target for passaging.
So you don't want them bumping into each other too much.
Yeah, exactly.
Okay.
That makes sense.
But cells need more than just space, right?
Oh, for sure.
Don't they also need the right nutrients to thrive?
They absolutely do.
Okay.
Just like cells need a balanced diet,
Mm-hmm.
and hybridoma cells are particularly fond of glucose and glutamine.
Oh, interesting.
These nutrients can get depleted pretty quickly
Yeah,
in a crowded culture.
right.
That's why regularly checking and replacing the cell culture medium,
Mm-hmm.
essentially their food,
Right.
is essential to keep those antibody factories humming.
Okay, so we've got our cues
Right.
for when it's time to passage these cells.
Yep.
But what does the actual process look like?
Okay, so
Walk us through the steps of giving these cells a new home.
the first step is to carefully check the cell density and viability.
Okay.
We can use a special counting chamber called a hemocytometer
Mm-hmm.
or an automated cell counter for this.
Got it.
You want to make sure a good percentage of the cells are alive and kicking
Right.
before you proceed.
Makes sense.
Then, depending on the density,
Okay.
you either dilute the culture by adding fresh medium,
Mm-hmm.
or you split the culture, transferring some of the cells to a new flask.
Oh, okay.
It sounds like handling these cells with gentleness is crucial.
Yeah.
You don't want to stress them out during the move.
Exactly. So once the cells are settled in their new home,
Yeah.
they're placed back in an incubator,
Right.
which is like a climate-controlled apartment for cells.
Exactly.
Kept at a cozy 37 degrees Celsius with just the right amount of carbon dioxide. Wow, it's amazing how much care goes into maintaining these tiny antibody powerhouses.
It really is.
But I imagine things can go wrong sometimes, even with the best care.
You're absolutely right.
Oh, no.
Hybridoma cell culture can be quite delicate,
Yeah,
and even experienced researchers encounter challenges from time to time.
I bet.
We'll dive into those challenges and how to troubleshoot them in the next part of our deep dive.
I'm already eager to hear more about that. Stay tuned as we continue exploring the fascinating world of hybridoma cells. So before we were talking about how even with the best care,
Right.
hybridoma cells can sometimes throw us a curveball.
They can.
So let's get into the detective work.
Okay.
What are some common hiccups
Yeah.
researchers encounter?
One common issue is low cell viability,
Okay.
where A significant portion of the cells just aren't thriving.
Right.
It's like a plant wilting
Okay.
because it's not getting enough water or sunlight
Right.
in hybridoma cell.
Yeah.
This can be caused by things like nutrient depletion,
Okay.
overcrowding,
Mm.
or even changes in pH or temperature.
So it's like a microscopic version of location.
Absolutely.
Okay. What are some strategies for addressing this?
The first step is to investigate the possible causes.
Okay.
For example,
Mm.
if nutrient depletion is supplementing the media with additional nutrients
Okay.
might be the solution.
Got it.
If overcrowding is the culprit,
Yeah.
passaging the cells more frequently
Okay.
can give them more space to thrive.
Right.
It's all about carefully observing the cells
Mm-hmm.
and making adjustments to their environment.
Fascinating.
Yeah,
So it's like being a detective,
right.
but for tiny cells.
Exactly.
Okay, what about other challenges? What if the cells are alive,
Sure.
but aren't producing enough antibodies?
That's another common issue.
Yeah.
And it can be quite frustrating.
Right.
It's like having a factory that's up and running,
Mm-hmm.
but not producing enough product.
Right.
One possible culprit
Yeah.
could be overgrowth.
Okay.
Even if the cells seem healthy,
Right.
if they get too dense,
Yeah.
their antibody production machinery
Mm-hmm.
might not function at its peak.
So even though we want them to multiply,
Right.
we also need to keep their population in check.
Exactly. Maintaining a slightly lower cell density
Okay.
can sometimes boost antibody production.
Gotcha. So,
Another thing to consider
yeah.
is the composition of the culture medium.
Okay.
Hybridoma cells are picky eaters.
Right.
And if they're not getting the right balance of nutrients and growth factors,
Mm-hmm.
their antibody production can suffer.
It makes you appreciate the complexity of these tiny cells.
It really does.
They're like high maintenance,
Right.
but incredibly valuable factory workers.
That's a great analogy.
Okay.
And speaking of keeping them happy,
Mm-hmm.
contamination is another challenge
Right.
you need to watch out for.
Oh.
Just like unwanted guests at a party,
Right.
Bacteria or fungi can wreak havoc in a hybridoma cell culture,
Oh, no.
competing for resources
Hmm.
and potentially harming the cells.
Contamination, that sounds like a nightmare.
It can be.
How do researchers prevent that from happening?
Sterile technique is absolutely crucial
Okay.
in hybridoma cell culture.
Yeah.
Everything from the lab equipment
Mm-hmm.
to the culture media and even the air in the lab
Wow.
needs to be kept as free from contaminants as possible.
Great.
It's like working in a miniature clean room.
Wow. It sounds incredibly meticulous.
It is.
But what happens if, despite all these precautions,
Right.
contamination does occur?
Well, that's when we have to act quickly
Okay.
to contain the problem.
Yeah.
Often this means discarding the contaminated culture
Oh, wow.
and starting fresh.
Okay.
It's a setback,
Right.
but it's better than risking the contamination spreading to other cultures.
Yeah. I can only imagine the frustration.
Right.
So we've talked about low viability,
Hmm.
decreased antibody production.
And contamination.
Yep.
Are there any other common challenges?
Yes. One more we often encounter
Okay.
is excessive clumping.
Okay.
Sometimes the cells get a little too friendly
Mhm.
and start sticking together,
Right.
forming clumps that can interfere with their growth
Okay.
and antibody production.
Right.
It's like a cellular traffic jam.
Okay, so what can be done about that?
There are a couple of strategies we can use.
Okay.
Gently pipetting the cells up and down
Mm-hmm.
can sometimes break up the clumps.
Okay.
Also, passaging the cells more frequently
Right.
can help prevent them from getting too crowded and clumping together in the first place.
So keeping them well spaced
Exactly.
and gently persuading them to separate if they get too cozy.
Right. Troubleshooting in hybridoma cell culture
Yeah.
is often a process of elimination and careful observation.
Okay.
We need to consider all the possible factors
Right.
that might be affecting the cells
Mm-hmm.
and make adjustments accordingly.
Now, even with these basic techniques, maintaining healthy, productive hybridoma cultures
Right.
can be challenging.
I bet.
But researchers have developed more advanced strategies
Okay.
to optimize these tiny antibody factories further.
That's what I've been waiting for.
All right.
Tell us about these advanced techniques.
One technique is using conditioned media.
Okay.
This involves taking the media from a healthy,
Mm-hmm.
thriving hybridoma culture,
Okay.
the leftovers, so to speak,
Right.
and adding it to a new culture.
Okay.
It might seem a bit strange,
Yeah.
but it actually can help the new cells adapt more quickly
Oh, wow.
and start producing antibodies sooner.
That's fascinating.
Yeah.
It's like giving the new cells a taste of success.
Yeah.
A little inspiration from their elders.
Right.
I'm curious, is there any research into why this works?
Yeah.
What's in that conditioned media that makes such a difference?
Researchers believe that conditioned media contains
Okay.
not just nutrients, but also signaling molecules secreted by the healthy cells.
Interesting.
These molecules might act like messages,
Okay.
telling the new cells to get busy producing antibodies.
Oh, wow.
We're still unraveling the exact mechanisms.
Yeah.
But it highlights the complexity of cell communication and the subtle ways we can influence cell behavior.
It's amazing how much we're still learning about these tiny cells.
It really is.
What other advanced techniques are out there?
Another important one is antibody titer monitoring.
Okay.
This involves regularly checking the concentration of antibodies produced by the hybridoma cells
Right.
using techniques like ELISA.
Okay.
That stands for enzyme-linked immunosorbent assay,
Right.
or Western blot.
Okay.
These techniques allow us to see if the cells are actually producing the desired antibodies
Got it.
and how much. It's like a quality control check for our microscopic factories.
So it's not enough just to keep them alive and well fed.
Right.
We need to make sure they're delivering the goods too.
Ex-
And speaking of delivering the goods,
Yeah.
what about when we need to produce large quantities of antibodies?
Right.
How do researchers scale up hybridoma cell cultures?
That's where things get really interesting.
Yeah.
When we need to go from a small-scale operation
Right.
to a full-blown antibody production line, we turn to bioreactors.
Okay.
These are specialized vessels designed to maintain optimal conditions for cell growth and antibody production at much larger volumes.
So we're trading in our handcrafted tools
Right.
for some high-tech machinery.
Exactly.
What are some key features of these bioreactors?
Well, bioreactors allow us to precisely control crucial factors
Okay.
like temperature, pH,
Okay.
oxygen levels, and nutrient supply,
Got it.
ensuring the hybridoma cells have everything they need to thrive
Right.
and produce antibodies efficiently,
Okay.
even at large scales.
It's like creating a miniature,
Exactly.
perfectly controlled ecosystem for these cells.
And there are different types of bioreactors,
Yeah.
each with its own pros and cons.
Right.
For example, stirred tank bioreactors use impellers, like tiny propellers,
Oh, wow.
to mix the culture,
Okay.
while airlift bioreactors use bubbles of air or oxygen to create circulation and provide oxygen.
Got it.
Choosing the right type of bioreactor depends on factors,
Okay.
like the specific hybridoma cell line,
Right.
the desired scale of production, and the sensitivity of the cells to shear stress.
Okay.
That's the force created by the mixing.
Gotcha. It sounds like a lot of expertise goes into designing and operating these bioreactors.
It definitely does.
Yeah.
Scaling up hybridoma cell cultures isn't just about having the right equipment.
Right.
It also requires a deep understanding of cell behavior and the ability to fine-tune the culture conditions
Yeah.
to maximize antibody yield and quality.
Welcome back to the deep dive. We've been exploring this incredible world
It is.
of hybridoma cells. These tiny antibody factories that play such a huge role in research,
Right.
medicine, and diagnostics.
Absolutely.
It's amazing how much ingenuity and precision go into harnessing their power.
It really is. From the initial cell fusion
Right.
to the meticulous care and the advanced techniques we've discussed, it's a testament to the dedication and creativity
Yeah,
of the researchers working with these remarkable cells.
absolutely. So thinking back on everything we've covered,
Correct?
what stands out to you as the most important takeaway?
I think the biggest takeaway is the sheer impact.
Yeah.
Hybridoma technology has had
Right.
monoclonal antibodies produced by these tiny cells have truly revolutionized medicine.
Oh, wow.
They're used in everything from diagnosing diseases
Right.
to treating cancers and autoimmune disorders, and they're even being explored for use in fighting infectious diseases
Right.
like COVID-19.
It's incredible to think that something so small
It is.
can have such a huge impact on human health.
Very-
And it all starts with understanding how to care for these cells, right?
Yeah, exactly. We talked about the basics of passaging,
Right.
the importance of providing the right nutrients and growth factors, and the constant vigilance needed to prevent contamination.
Right.
It's a meticulous process
Yeah.
that requires both scientific knowledge and a bit of an artistic touch.
You mentioned an artistic touch.
Yeah.
What did you mean by that?
Well, even with all the protocols and guidelines, there's still an element of intuition and experience.
Okay.
involved in working with hybridoma cells.
Right.
Sometimes you just have a feeling that something isn't quite right with a culture, even if all the numbers look good.
Right.
It's about observing the cells closely, noticing subtle changes and making adjustments based on what you see.
So it's like being a detective,
Right.
but also a bit of a gardener,
Yeah.
tending to these microscopic crops.
A great analogy.
We also discussed some of the more advanced techniques being used to optimize hybridoma cultures, like conditioned media and scaling up production in bioreactors.
Yeah.
It seems like the field is constantly evolving.
It absolutely is.
Okay.
Researchers are always looking for ways to improve antibody yields, increase efficiency, and reduce costs.
Makes sense.
And with new technologies and insights emerging all the time, the future of hybridoma technology is incredibly exciting.
What are some of the areas you're most excited about in terms of future research and applications?
One area that's particularly interesting is the development of new bioreactor designs and technologies as we move toward personalized medicine.
Right.
And the need for smaller batches of highly specific antibodies.
Okay.
Researchers are exploring miniaturized bioreactors.
Interesting.
And even microfluidic systems that can produce antibodies.
Wow.
On a tiny scale.
So even smaller than the tiny factories we've been talking about.
Exactly. These micro-scale systems could revolutionize antibody production, making it faster, more efficient, and more customizable.
That's incredible.
Another exciting area is the use of genetic engineering techniques to further enhance hybridoma cell lines. Researchers are working on ways to modify these cells to produce antibodies with even greater specificity, potency, and stability.
It's amazing to think that we can engineer these cells.
It really is.
At such a fundamental level.
It really highlights the incredible power of biotechnology. And it underscores the importance of continued research and exploration in this field. Who knows what amazing discoveries and applications lie ahead?
Well, one thing's for sure, this deep dive has opened my eyes to the incredible world of hybridoma cells and the crucial role they play in advancing human health and scientific understanding.
Absolutely.
To our listeners, we encourage you to keep that curiosity alive. Delve deeper into this fascinating world of cells and biotechnology. You never know what amazing discoveries you might uncover.
And remember, the next breakthrough in hybridoma technology could come from anyone with a passion for science and a willingness to explore the microscopic wonders of life.
That's all for this deep dive into the world of hybridoma cells.
Thanks for listening.
Thanks for joining us.